In 2025, a sealed dossier emerged from deep within the Royal Archives, and inside, non-destructive DNA tests on Queen Charlotte's personal effects hinted at ancestry and health secrets the British establishment long avoided. The evidence was so controversial, it was nearly buried for good. What could be so disturbing that experts feared its release? Now, for the first time, the full story is ready to be examined. The dossier, dated March 2025, catalogued a collection of Queen Charlotte's personal effects that had quietly passed through generations of royal custodianship. Each artifact carried a meticulous accession record, tracing its journey from the private chambers of Kew Palace to the climate-controlled vaults of the Royal Collection Trust. Among the items listed was a hand-stitched linen handkerchief, a silver-capped cosmetics jar bearing faint traces of powder, several sewing needles stored in a mother-of-pearl case, and a bundle of correspondence sealed in brittle wax. The inventory also included a fragment of silk from a court gown, its fibres preserved in an archival box since the late 1700s. A senior archivist responsible for overseeing the Queen Charlotte holdings recounted the strict protocols governing access to these materials. Each transfer between institutions, whether for exhibition, conservation, or research, required dual signatures and a photographic log. The 2025 review introduced a new layer, a replication clause that mandated any biological sampling be performed in duplicate, with parallel samples sent to an independent laboratory for verification. This measure, rarely invoked in royal artifact research, underscored the sensitivity of the project. Chain of custody records documented every moment the artifacts left secure storage, noting the presence of conservation staff, external scientists, and security personnel. The archivist described how the sampling team prepared a detailed logbook, recording the precise time, method, and personnel involved in each action. With these controls in place, the dossier established an unbroken provenance for each sampled object, setting the stage for a scientific inquiry that would test the boundaries of both archival practice and royal discretion. A forensic environmental DNA scientist described the laboratory's approach to sampling Queen Charlotte's effects, emphasizing the importance of non-destructive methods. Each artifact was handled with gloves, and only sterile, ultraviolet irradiated swabs or electrostatic lifts made contact with the surfaces. The team avoided any abrasion or wetting that might alter the original fabric or leave visible marks. For the linen handkerchief and silk gown fragment, a single sterile swab was gently rolled across the least conspicuous edge, then immediately sealed in a barcoded tube. For the cosmetics jar and sewing implements, adhesive film lifts were applied for less than 15 seconds, then removed and stored under controlled conditions. To guard against contamination, every sampling session included blank controls, unused swabs, and adhesive films processed alongside the real samples. These blanks were tracked using the same chain of custody protocols as the artifacts themselves. The laboratory also processed blind samples, some containing only buffer and others spiked with trace amounts of known DNA, to monitor for cross-contamination or procedural errors. After extraction, the DNA fragments were measured for length and for characteristic damage patterns. Authentic ancient DNA from these surfaces typically measured between 35 and 70 base pairs, with a distinct signature of cytosine to thymine changes at the fragment ends. Any sample lacking this pattern or showing fragment sizes typical of modern DNA was flagged for further review. By combining surface-only recovery, rigorous negative controls, and molecular authentication, the team established a standard of evidence designed to withstand the highest level of scientific scrutiny. This meticulous process set the foundation for any subsequent genetic findings. A population geneticist reviewed the first sequence data from the artifact swabs, focusing on both mitochondrial and autosomal markers. The mitochondrial DNA, often used to trace maternal heritage, produced a match rate that was neither definitive nor easily dismissed. The resulting haplogroup signal hovered between Mediterranean and Atlantic populations, broad regions that include parts of southern Europe, North Africa, and the western coasts of the Iberian Peninsula. With coverage limited by the fragmentary nature of ancient DNA, the geneticist cautioned that these results could only be described as suggestive. Statistical confidence intervals stretched wider than would be acceptable for modern forensic work, leaving room for ambiguity. Autosomal markers, which reflect the full spectrum of ancestry, echoed the mitochondrial uncertainty. The data hinted at a blend not fully accounted for in established royal genealogies, but the signal was diluted by low red depth and the inherent challenges of working with centuries-old samples. The geneticist emphasized that while the ancestry component appeared unexpected in the context of British royal records, the evidence could not be called conclusive. The findings were logged as a possible anomaly, pending further biochemical and proteomic analysis that might clarify or complicate the emerging picture. Mass spectrometry of the artifact residues revealed a suite of peptides closely associated with inflammatory processes in human tissue. Among the fragments, researchers identified sequences matching alpha-1 antitrypsin and C-reactive protein, both markers that in modern clinical settings indicate chronic activation of the immune system. Such elevated peptide signals are rarely preserved in textiles and personal effects, suggesting repeated contact with biological fluids over extended periods. Chemical analysis of the cosmetics jar and powder residues added a further layer of complexity. Trace amounts of mercury and lead compounds were detected, consistent with ingredients commonly found in 18th century face powders and skin lighteners. Mercury sublimate and white lead were favored for their whitening effect, and they were widely used despite their documented toxicity. Repeated exposure to these metals is now known to trigger or worsen immune dysfunction, often manifesting as swelling, joint pain, and persistent fatigue. The convergence of inflammatory peptides and heavy metal traces in Queen Charlotte's personal items points toward a pattern of physiological stress. The evidence suggests not only ongoing immune activation, but also a possible chemical burden from daily cosmetic routines. These molecular findings offer a biomedical context for symptoms described in historical accounts, raising new questions about the interplay between environmental exposure and chronic illness in the royal household. Subscribe for more groundbreaking historical investigations. A medical historian reviewed the handwritten records from Queen Charlotte's final years, noting repeated references to swelling in her legs and face, persistent fatigue, and a steady decline in her ability to walk. The term dropsy appeared often in the physician's notes, a catch-all diagnosis for fluid retention that rarely explained the underlying cause. Modern analysis of these symptoms, especially when paired with biochemical evidence of chronic inflammation, suggests a pattern more consistent with autoimmune or inflammatory disease than with simple heart failure or age-related decline. The historian traced the gradual progression. Letters from the 1790s described Charlotte's increasing difficulty standing unaided, her reliance on attendants, and her withdrawal from public life. Family correspondence mentioned her frustration with constant discomfort and unpredictable bouts of pain. These lived experiences, recorded in the Queen's own words and those of her caregivers, echo the molecular findings from her personal effects. The convergence of historical testimony and scientific data invites a reconsideration of what was once dismissed as an inevitable consequence of old age. Subscribe for more groundbreaking historical investigations. An internal memo circulated among senior curators and research leads in April 2025 outlined the dilemma facing the Royal Collection Trust. The document, stamped confidential, referenced the preliminary genetic findings and the potential for public controversy if signals of ancestry and health markers were released without further verification. Citing ethical guidelines established by the International Council of Museums, the memo recommended a temporary delay in publication and emphasized the need for sensitivity toward living descendants and the broader implications for public understanding of royal heritage. It described concerns about misinterpretation and the risk of fueling speculative narratives, especially given the ambiguity of the initial data. In response, the Trust convened a roundtable with external geneticists and independent historians, aiming to balance scientific transparency and institutional responsibility. The roundtable minutes recorded a spectrum of opinions, from calls for immediate disclosure to arguments for extended peer review and descendant consultation. This debate, documented in formal correspondence and meeting logs, underscores the tension between advancing knowledge and respecting the weight of historical legacy. Subscribe for more groundbreaking historical investigations. Science keeps rewriting royal histories, one molecule at a time. Today, our understanding of identity, ancestry, and inherited illness is far from settled. As new evidence emerges, the past grows less distant, and the questions we ask about power, legacy, and truth become more urgent. Subscribe for more groundbreaking historical investigations.